Welcome everybody to the NCE6. So this session, the title is Translating Tissue Engineering from Benchtop to Chairside. And today we're really privileged to have two giants in the field who really walked the journey from bringing innovative ideas in research all the way through regulatory to real-world evidence to drive clinical applications. So many of you in the audience, you're clinicians practicing either full-time periodontist, you may be in academia doing these innovative research, or a combination of both. So today the session is really telling you a story and illustrating a journey where you have an innovation in biologics or biomaterials, and how do we work together as a community to drive these changes to real-world impact for our patients. So we're very excited to have Dr. Sam Lynch together with us today, as well as Dr. Michael McGuire, and they will be really showcasing that journey from discovery all the way to clinical impact. So without further ado, the order of the presentation will be Dr. Sam Lynch first and Dr. Mike McGuire second. So introduction for Dr. Sam Lynch. Dr. Sam Lynch really needs no introduction, but he's a pioneer in regenerative medicine, really at the birth of regenerative medicine and the concept of tissue engineering that was coined in the 1980s, 1990s by Bob Langer at Massachusetts Institute of Technology in Boston. And he's one of the world's most foremost authorities on growth factors for tissue engineering and regeneration. 250 publications, patents, given hundreds of lectures over 25 countries, and his particular innovation is the pure recombinant platelet-derived growth factor and specific biomaterials. So he has conducted several pivotal clinical studies and carried these through the FDA 4 times in various applications, from not only dentistry and periodontal regeneration, but also orthopedics, and more recently, skin rejuvenation and regeneration, and also diabetic wound healing. So, as periodontists, it's important for us to know our positioning as oral health professionals, but also in the global context of medicine and healthcare. So he's gonna tell you a little bit more about his journey, and right now he's the founder and CEO of Lynch Regenerative Medicine, focused on bringing these revolutionary regenerative technologies to more patients and more clinical indications. So without further ado, let's give a warm round of applause for Dr. Sam Lynch.
Hey. Thank you, David. All right, thank you, David, very, very much. It's great to see you all this morning. I had the good fortune of doing a lot of different things in my career, in orthopedics and wound healing and diabetes and more recently aesthetics. But I'm a periodontist, and it always feels like coming home to come to the AAP or just to lecture to any perio group. So I'm so excited to be here, and I hope you'll learn at least something this morning in the next 20 or 25 minutes. I promised Mike I wouldn't take too much of his time. This is my disclosure. So yeah, let's talk about some growth factors. As you guys know, there's lots of different growth factors, right? They all do some things in common, but they all do them very, very differently, and some do some things better than others. So there's insulin-like growth factors, epidermal growth factor, fibroblast growth factor, transforming growth factor beta and alpha. EPO is a growth factor. There's nerve growth factors, right? But there's only been one growth factor that has ever gotten FDA approval for tissue regeneration, and that is platelet-derived growth factor. And so, as David said, that's where I spent most of my career, and that's where I'll focus my presentation this morning. So PDGF, platelet-derived growth factor, it is one of the most important, if not the most important, agents, proteins in our bodies naturally to stimulate healing. It is incredibly highly conserved protein. What does that mean? It means that the PDGF in mice and the PDGF in humans are essentially identical. You can take human PDGF and put it on a mouse wound, and it'll stimulate healing of the mouse wound. You can take mice PDGF and put it on a human wound, and you'll get a little immunologic reaction, but it'll stimulate the healing of the human wound, right? It is so fundamentally critical to the very survival of our species, right, that has been conserved throughout evolution. And I like to say it is the most potent, the most sophisticated, the most complex, and the most effective drug delivery system that's ever evolved, that's ever been created. PDGF, right, is an incredibly potent mitogen. It stimulates cell proliferation. It's a potent chemoattractic agent. It stimulates cell migration, right? So if you had this incredibly potent growth factor just freely floating throughout your body, right, we might weigh 30 tons by the time we're 30 years old. So instead of that, what has evolved is this drug delivery system where this incredibly potent growth factor is contained in platelets. And what happens whenever you have an injury? Right? You have a blood clot that forms, whether that injury is from periodontal surgery or aesthetic surgery, plastic surgery, dermatology surgery, or an accidental injury, right? A blood clot forms. What's a blood clot? Simply a coagulation of platelets in a fibrin meshwork, right? And so those platelets, when they recognize the subendothelial tissue, the tissue surrounding the blood vessel lumen, they become activated, they stick, they form the clot. When they do that, they release PDGF and a few of the other growth factors exactly where you need it. This amazing drug delivery system, right? And then it stimulates all these great things to happen. I like to say, right, I spent my whole career looking at 3 very simple questions. How does the body know that it's been wounded? How does the body heal itself? And how does the body stop healing itself? Right? And we're going to talk about that as we go forward. Now, growth factors at this point have been around for 30 years, right? We started 30 years ago looking at blood and blood platelets and PRP. PRP's been around for a long time. But we recognized early on in the research lab that we could take, blood and platelets and PRP, and sometimes it would have a lot of growth factors and sometimes it wouldn't. So we were like, well, how do we really, right, want to develop a medicine that really works the same every time? And we were working across the hall from some insulin researchers, and they were wrestling with the same problem because at that point, if you remember, right, we used to get insulin from pig and bovine pituitary glands, right? So, right, they were like struggling with this. So working together, we came up with this ability to produce recombinant proteins. So you take the gene, you find it, you have to purify the protein. In the case of insulin, that was pretty easy, right? Insulin's a 7,000 kilodalton protein. PDGF is a 25,000 kilodalton protein that's a dimer, meaning it's 2 polypeptide chains linked together by disulfide bonds, right? So that took us years to just purify PDGF and get the amino acid sequence. But once we did that, then we were able to build molecular biology probes. We were able to probe the human chromosome, find the gene on the human chromosome that encoded for PDGF, clip that gene out using certain what's called restriction enzymes and other molecular biology techniques, and put that one gene into, in our case, we decided to use yeast, right? And so we put that one human gene in yeast. Now that yeast produces one human protein. In our case, PDGF. In the insulin researchers' case, it was insulin, right? So you can get an absolutely pure, consistent, sterile preparation of a product, of a growth factor, of a biologic using this technique. And now there are probably 100 different biological drugs, right, produced using this technique. Okay, that just talks about kind of how you go about kind of making, right, whatever active ingredient it is that you're interested in. Now you got to take that and you got to actually develop a medicine, something that's going to help people with it. And this is just a simple little chart, right, that talks about the FDA process. And so, if you're doing something that's, not too novel, it can get through using what's called a 510K process. That's roughly maybe $5 to $20 million in 2 or 3 years. If you're doing something a little more novel and more complicated, and it's a device or it's a drug device combination, right, that might take $50 to $100 million, right, and 5-6 years. And if you're doing a new drug from scratch, what we call an NCE, a new chemical entity, that may take many, many hundreds of millions of dollars and 8 to 10 years to do. Okay, so what's the first step in that process? The first step is, right, you have to prove safety. Everybody, first step in anything has to be, is this product, is this PDGF in our case, is it gonna be safe? We have spent like literally millions, tens of millions probably of dollars, I hate to think how many, right, proving safety just of PDGF. So we started out with cytotoxicity studies. We did dermal and ocular irritation studies. We did hypersensitivity studies. We did 8 mutagenicity studies. We did 4 acute systemic toxicity studies. We did 4 repeat dose toxicity studies. We did chronic tox studies. We did reproductive tox studies, and we did local implantation studies. We did intradermal injections, sub-Q injections, intraperitoneal injections, and intravenous injections, as well as, of course, surgical implantation. People think, right, oh, you probably just tested safety in the mouth. FDA doesn't let you get away with that, right? You have to show it's safe throughout the entire body, right, before they'll ever let you put it in humans. And then you have to do that on multiple dose levels. So we didn't do just 10x or 100x or 1,000x. We went up to 40,000 times the maximum therapeutic dose in these different graft materials. We did, again, single and repeated dose studies and all these different routes of administration just to prove safety of PDGF. Now, we got lucky. I mentioned we were unlucky on the manufacturing side and trying to purify and get the amino acid sequence of PDGF because it's such a complex molecule. But we did get lucky in one other way. It turns out because PDGF is so potent that our bodies have developed a regulatory mechanism. You know, I described for you earlier how PDGF is just delivered to sites of injury. Well, it turns out that if you apply PDGF or you inject PDGF intravenously, the body has a very potent, rapid regulatory mechanism to make sure that it's cleared from the body very quickly. So the half-life, if you inject PDGF intravenously, the half-life in the human body is about 2.5 minutes. So that means that every 2.5 minutes, half of the protein is cleared from the human body, right? So it makes it a really safe product to use locally. So these are the different FDA approvals, and I'm going to go through each of one of those in more detail. So we got 4 FDA approvals. It's taken us about $100 million of investment to develop these 4 different products. So the first one, as David mentions, in treatment of diabetic foot ulcers. We'll just talk briefly about that, right? Then we did GEM 21 for dental oral surgery, perio surgery applications. And more recently, then we did orthopedics, developed a bone grafting material for orthopedic applications, 2 different versions of that, one surgically implanted, one injected. And then more recently than that, we did topical aesthetic dermatology and plastic surgery indications. So this is from an article that we just recently got accepted into the Journal of Plastic and Reconstructive Surgery, looking at all the different medical therapeutic applications of recombinant pure PDGF. And I would refer you to that article if you're interested in more detail. Okay, so the first pure PDGF product was developed by Johnson & Johnson. I consulted with them, was to develop this treatment based on PDGF for skin diabetic foot ulcers. This was taken pure PDGF and just applying it in a collagen sponge twice, actually, to this DFU, right? GEM 21, I think you all know about that. Thank you, David. So this is, I like to say this is one of my cases. And when I say this was one of my cases, I mean this was my tooth treated by Dr. Mark Nevins, who's in the audience with me. So thank you, Mark, for this amazing, amazing case. So I had a mesial distal fracture of a lower first molar. And Mark can swear, this is honest God truth. So I did it. I knew when I did it. I went to the endodontist. He was like, you fractured through the fornix of the furcation. This is going to have to be removed, right? So like any good healthcare provider, I waited 6 months. Just because I knew I was going to be treated with PDGF, and I wanted to make it a little challenging. So here's what I presented to Mark with. I said, Mark, can you help me out? And Mark looked at this. He said, my God, where have you been for the last 6 months? So anyway, he treated it with recombinant PDGF, I think in a mineralized bone allograft. And you can see the amazing results that he was able to achieve, I'm sure because of the great surgical technique. This is an Isvan Urban case that was published in IJPRD, also using recombinant PDGF with BioOS and collagen membranes. And we can go into the details of these cases, but I'm just giving you examples here of different uses of PDGF in periodontal surgery. So for GEM21, focusing on that just for a minute. So how do we develop GEM21 specifically, right? So I actually started the research in 1985, 86, did 10 years of research at the bench, right? A lot of that molecular biology work I was describing earlier. I was a young peon in the lab, and so I basically had the 8 to 8 shift, meaning I started collecting fractions off the separation column at 8 at night and collected them till 8 in the morning. At any rate, going from that then, I went to launch BioOS and BioGuide in North America and South America and most of Asia for 5 years from 95 to 2000, and then started my own company called Biomimetic Therapeutic in 2001. It took us about 5 years then and about $25 million from the start of biomimetics to develop GEM21. The good thing about all of that is if it works, right, you can be very, very successful. So this is our IPO on NASDAQ. It's a great day. We went from there then on to developing augment bone graft for orthopedic applications. That was about 7 or 8 years and about $75 million to develop augment. Augment is a bone grafting material containing recombinant PDGF, primarily used as a substitute for iliac crest and proximal tibial bone grafting in orthopedic surgery. Okay, so then on to more recently, just published this article in the Journal of Cosmetic Dermatology, and it is looking at the use of the same pure recombinant PDGF that you have in GEM21 for topical facial applications. PDGF is a pretty amazing molecule, right? It stimulates both bone regeneration and soft tissue regeneration. I tell most people outside of perio that, and they don't believe me, right? It's like, how can one growth factor stimulate both bone and soft tissue? In perio, we know, right? Because we deal with bone and soft tissue together every day of our professional lives. Okay. So this is some uses of PDGF and topically in aesthetics. This is a patient who had microneedling, if you're familiar with that, in the face, and 1 hour post, right? So one of the things that's become really apparent in this aesthetic plastic surgery dermatology world is that topical PDGF will really, really dramatically reduce the inflammation of the patient. So this is a patient that had laser resurfacing of her abdomen, and you can see immediately post-laser and then 5 minutes post-laser, and you can see the dramatic reduction in the inflammation, the burning, the redness of that patient. Here's another patient before and 72 hours afterwards. Here's a post-inflammatory hyperpigmentation patient. Again, 1 hour, 24 hours, and 12 days. Here's a plastic surgery patient, right? The patient couldn't really get the neck wound to heal from the facelift and was treated with PDGF topically. Here's another facelift case, right, with recombinant PDGF. This is a patient who had pre-auricular scarring, so right in front of the ears where they do the incision for the facelift. She didn't like that. She wanted to have laser treatment to treat those scars. She did, and then the surgeon couldn't get the scarring to heal. the laser burn to heal. So she was then treated with PDGF, and you can see the result. Here's a combination treatment. So this patient had both a facelift and ablative CO2 laser skin resurfacing. So you can see the immediate post-op, 48 hours, 5 days, 9 days. This plastic surgeon said the results in 12 days are what we used to see in 3 to 4 weeks. So that's all the great stuff. But in the last 2.5 minutes, there are some challenges. So when I left academia, as I said, I went to help launch BioUs and BioGuide throughout most of the world. And we were just getting started, right, with a great, we did a lot of great studies. Dr. McGuire helped us do some studies, Dr. Nevins and so forth. We're really starting to get a lot of traction. And then what comes along, if you remember, in the mid to late 90s? Mad cow disease. Obviously, BioUs, and we use a lot of bovine-derived products, right? So we had to spend 2 years, right, showing that all these collagen products, specifically BioUs, right, didn't have any prions in them. We don't really talk about that much anymore. But that was a huge setback. You can imagine when you start seeing it on the 6 o'clock news, that's not a good thing, right? Number 2, another challenge, the golden rule. What's the golden rule? It's not the golden rule you're thinking of. Golden rule is the people with the gold make the rules. That's what happens when you do translational medicine, right? This, again, it costs millions or hundreds of millions of dollars to do these things. And you need to get investors to do that. You don't get a blank check, right? It always comes with strings attached. Oh, this is a great story. Adverse events. So we had just gone public. We were a public company on NASDAQ. We were feeling great, right? We were doing a groundbreaking for a new facility. I'm up there kind of giving some little speech, probably boring everybody to death. And all of a sudden I see my management team start to talk to each other, and then they're running into the building, and I'm like, what's going on? So this whole presentation, the groundbreaking, I'm like, what is going on? So it turned out, right, that the FDA had made an announcement about PDGF for the diabetic foot ulcers, that long-term use of PDGF, right? And because we were using PDGF for other medical therapeutic applications, totally different applications, totally different formulations, but our stock price in the time, I have to say, this is the most expensive lecture I've ever given. And that 15-minute lecture talk, whatever, our stock price dropped $150 million in valuation. That's not a good day. All right. And then, right, if you're in, and I'm about finished, Mike, I promise. Then if you're fortunate enough, you get great randomized controlled clinical trial data, phase 3 data, you take it to the FDA. They're like, okay, looks okay. Well, let's go do, an FDA advisory panel. Again, you've heard, right, on the news and so forth, different, new medicines, they go to the FDA advisory panel. So an FDA advisory panel, in our case, we had 18 physicians on the FDA advisory panel. This was for our first orthopedic product. I was told at the time it was the largest FDA advisory panel since breast implants, silicone breast implants, right? So at the end of this day, it's an 8-hour day. You start at 8 in the morning, you finish at 5 at night, right? And you get a thumbs up or a thumbs down vote, right? They shut our trading down on NASDAQ, right? So we didn't trade the whole day because they knew everybody's going to be trying to read the tea leaves. It's going to get approved, it's not going to get approved. Because if it doesn't get approved, guess what? You just spent that $75 million for zero. Right? Literally for zero, because if it doesn't get approved, it's not worth anything, right? So all day, everybody's trying to guess, is it going to get approved or not, right? There's been articles written about this. So at the end of the day, FDA was very lukewarm to negative, but all of our orthopedic surgeons that participated in the trial were incredibly enthusiastic and positive, because it meant they didn't have to drill into the iliac crest, into the hip, right, to get autogenous bone. They could just take a product off the shelf. Anyway, we ended up winning, 10 to 8. One doc changed his vote at the last minute to a positive, and that was the difference. That was a difference between a $75 million total loss, and we ended up selling the company for like $450 million, all based on one vote in one day. So I leave you with that message. Translational medicine is awesome. I love it. I spent my whole career doing it. I wouldn't have done anything else, but it is not for the faint of heart. Thank you very much.
So thank you so much, Dr. Lynch, for your insight. And I think everybody thinks, the path of translation, it's simple. You follow the FDA, you get it approved, but you don't really see the behind the scenes, right? I would love to, what were you feeling in those moments? How did you overcome challenge? So maybe that's what we're going to talk about afterwards. The next speaker is Dr. Michael McGuire, and his topic will be really bringing real-world evidence in clinical trial. When something gets approved, for all the clinicians in the room who want to utilize the technology, you want to see evidence, you want to see long-term follow-up. How do we do that consistently? So Dr. Michael McGuire is the author of over 85 peer-reviewed publications in scientific manuscript, founder and chairman of the McGuire Institute, a nonprofit organization that promotes practice-based research. And that's his life mission, to bring evidence into the clinical practice of periodontology. His research has received numerous awards, including the American Academy of Periodontology's highest recognition of excellence, 4 times, including the Earl Robinson Regeneration Award, twice the Clinical Research Award, and he's also the recipient of the Master Clinician Award from the American Academy. And he's also somebody that gave back to our academy in numerous leadership roles, including the AAP president, as well as the president of the AAP Foundation. So let's give a warm round of applause to Dr. Michael McGuire.
Thank you. Thank you, David, for the nice introduction, and the organizing committee for the invitation, and all of you for coming to this lecture. As I had not obviously heard Dr. Lynch's presentation, and as I was thinking about, what I was going to say, one of the things I was going to say was, thank you, Dr. Lynch, for a great presentation, and it was, but I was going to say, and he makes it look easy, doesn't it? There wasn't anything easy about that. This is tough business, and that's kind of what we're going to talk about here on how you get through this process and all the way to adoption. We have to provide our conflicts of interest, and as it relates to this particular lecture, I really have none other than the fact that I am founder and chairman of the McGuire Institute, and in this case, I'm going to, during this lecture, be using the McGuire Institute as an example of practice-based research. So, it's a process, and we want to be able to drive these innovative clinical therapies, like Dr. Lynch was talking about, toward adoption. Adoption's really the key. And if you're a researcher and you have some really promising early results, fantastic, congratulations. If you're able to get through the ADA, a fabulous milestone. You got to do it, wonderful. But the way I look at promising early results and FDA approval, they are the table stakes. They're the ante in the poker game. They're the bare minimum necessary for you to sit at the table and play. And the results of all of your efforts are going to be dependent on all the things I'm going to be talking about in my lecture. All of the things that are going to drive those dentists down to your booth at the exhibit hall, and even more importantly, provide things that your patients are really going to value in the clinic chair. One of our problems is something called translational lag. The authors of this paper out of the basic science journal Science, they looked at 4 years of basic science journals, and they identified 100 discoveries with clear clinical potential. And they followed those 100 discoveries, and what they found was, out of 100 discoveries with clear clinical potential, only 1 achieved widespread adoption, and it took 15 years. If you have a company, you don't have 15 years to make this work. And as they tried to determine why is that, one of the suggestions was that they suggested that it's really critical to have multidisciplinary collaboration in these projects, interacting with scientists and clinicians. The NIDCR found a similar thing when they did a retrospective kind of look back. They said, okay, we've spent $100 million over the last 10 years. Out of that $100 million, how much of that really went to creating meaningful outcomes for the patients? And what they found was very little. And that's a problem. I mean, how can you spend 10 years of time, $100 million, and not really get anything out of it? So obviously the next question is why and what can we do about that? And what they found was they were funding all of these basic science projects. The basic scientists were doing their favorite science project in a silo, barely talking to each other, much less with clinicians. And once they finished, they threw it over the fence to the clinicians, and the clinician says, what's this? It's an answer to a problem I don't have. And so the NIDCR said, okay, we can't have that in the future. That if you're going to want NIDCR funding, or at least some help, you're going to have to demonstrate how you're going to involve clinicians earlier in the process than you've had in the past. And there's no better way to do that than to be involved with a practice-based research group. In our case, the McGuire Institute. I founded that, and I've been involved in practice-based research since 1989, but founded IMAC in 2010 as kind of a legacy project to help train other like-minded full-time private practitioners that have an interest in being able to produce high-level research in the midst of a busy practice. It's not an easy thing, and so we help train those individuals. We have 17 different sites, as you see across the map, with 28 different investigators, and we purposely have kind of different age groups of investigators at these sites. We've got senior people like me and mid-career people and rising stars, so that hopefully it's going to be evergreen as we move forward. And we not only train the investigators, but also their auxiliaries on how to be able to do this, because you have to have the infrastructure there. And so what are the unique things that practice-based research brings? Well, there's a number of them. And the first thing is more rapid enrollment and more rapid completion. How do we do that? Well, the patients or the subjects in our studies come from our offices. These are our patients. So we're not having to advertise for them. We're not having to go out and induce them to come in somehow. And for that reason, we generally can be much more rapid enrollment, which obviously leads to more rapid completion. In addition, we can be more efficient. We try to batch. If we have a study day or a study afternoon or a morning, we're batching study visits, we're batching surgeries, and we are able to get fast IRB approval, which is a huge thing. I mean, if you're working in a university, you know how difficult that is. I mean, we get IRB approval in 2 weeks because we're working with private IRBs. They're just as rigorous. We're not skipping any steps. We're just skipping the bureaucracy, and time is money. In addition to that, practice-based research allows us to have less follow-up. I was at a study meeting this morning, and we saw that one of our studies we're just completing, we had virtually no loss to follow-up. And why is that? Okay, again, because these subjects are our patients. We have relationships with these people. They're not just floating in or out of the clinic. They're staying there. And by not having loss to follow up, it does a lot of important things. All of you know that we power our studies based on statistical claims that we want to make. And if you lose patients, you lose your ability to make those statistical claims. You can ruin the study. In addition to that, just almost as important, is it allows us to have long-term follow-up studies. You know, I've been in practice and full-time private practice for 45 years. Everything works at 6 months. You know, I want to know what happens at 1 year, 3 years, 5 years, or even longer. Here's 2 studies that we've done, one 6 to 8 year follow-up, another at 10 years. And it's very difficult to get that kind of long-term evidence if you're not doing practice-based studies, because since these people are in our practice, we can continue to follow them. In addition, if you're a study sponsor, this is tremendously important information at a really low cost, because it's just one more study visit. And so we write all of our protocols anytime that we can to include long-term follow-up. Also, it provides treatment that takes place in a real-world environment in patient populations that are representative of private practices, resulting in evidence that's more generalizable to clinical practice. And that's huge. You don't want your studies done in an environment that could never be duplicated in a practice, or on patients that look nothing like the patients that you want to have your products used on. Very important, and ultimately that leads to providing more rapid translations of new therapies to clinical practice. The FDA now has something called the Real-World Evidence Program. And they mandate now, if you're going to run a study through the FDA, you have to demonstrate how will your results produce real-world evidence. Why have they done that? Well, they have found that many of the studies that they've done over the last decade, lots of implant studies, the results do not reflect what is really happening in the real world. And so they are demanding for you to demonstrate how that's not going to happen. And there's no better way to generate real-world evidence than practice-based research. And how do we, as a not-for-profit, do it? We have 28 investigators with over 400 years of combined experience in 17 private practices, 15 general dentists at 12 sites who serve as sub-investigators. Our investigators have more than 1,500 different doctors who refer to us. They perform hundreds of patient procedures a day, and within our network, there are more than 30,000 patients on maintenance programs with 300 hygiene appointments a day. So lots of opportunities there to get practice-based evidence. And our investigators have published more than 600 peer-reviewed papers. And I want to share with you a few of these papers that are part of the PDGF story. You know, I was fortunate to be one of the investigators on Dr. Lynch's pivotal trial when PDGF became FDA approved. He, as usual, was ahead of his time in that he created a study here that was multi-center with university sites and practice-based sites. And I think you'll see that paid off in the way that we were able to manage these publications. And here, this is one of my cases that I continue to follow, but it was in the study where you can see some amazing supracrestal bone growth there with the PDGF. So following the pivotal trial, we then worked with Dr. Giannobili and did a quick study for the scientists on wound repair. And if you do one for the scientists, you got to do one for the clinicians. And here I partnered with Rich Gao, again, another practice-based study where we're looking at both clinical and radiographic analysis from 24 to 36 months. This is important information and showcasing the importance of practice-based research. And Sam, another thing, he was pretty lucky with his initial FDA approval. He got pretty broad application where he got not only hard tissue approval, but soft tissue. And so my recollection as I went to Sam, I think it might have been at a meeting here, and I remember in the hall meeting up with Sam. You know, I was an aggressive young guy, you know, and I said, you know, Sam, you've got bone studies, but you have approval for soft tissue, but you don't have anything. Let's do something. And so, you know, the way we do these things is we almost always set up a pilot. So here was the pilot, and we're going to compare coronally advanced flap with PDGF to the gold standard connected tissue, and I was able to show proof of principle. Well, once you show proof of principle, then you need to move into a randomized controlled clinical trial. And so here is, we did a RCT looking at connective tissue graft versus PDGF with coronally advanced flap, and we also looked at micro-CT and human histology. We'll talk about that in a moment. But with the RCT, we were able to show equivalence in root coverage between the gold standard and this new therapy. And the pros, the patient-reported outcomes, were more positive for the PDGF because, of course, there's not a donor site. And what do? We're going to follow that long term, right? So here are all of our patients at 5 years. No significant difference between baseline and 5 years for connective tissue graft versus PDGF. And if we go back to the micro-CT and the human histology, This award-winning publication demonstrated that, yes, you can get, we showed with our RCT that you can get equivalent root coverage, but we showed with the human histology that with connective tissue graft, you're only going to get a soft tissue adaptation to the root surface, where with the PDGF, we're getting true periodontal regeneration. You have new cementum, new bone, functionally oriented PDGL. And so it is a concrete reason for the clinician to consider moving from a gold standard to an alternative. And this provides important information for the clinician. And unfortunately, it's an ongoing, never-ending thing. You've got to have, to move these things toward adoption, you've got to have the information out there, at least for the periodontists. And it's kind of a never-ending process to keep it top of mind. So, along with the publications, our investigators are out there talking about what they do. That our group, on the average, has 100 regional lectures, 55 national lectures, 20 international lectures. They're active at all levels of organized dentistry. Many are editors or serve on editorial boards of peer-reviewed, high-impact journals. And what that means is they're oftentimes invited to major podiums to be able to disseminate this information. And many are speaking at this meeting itself. But as Sam and I were talking before the meeting, podiums are starting to decline a little bit as far as the importance. This newer generation fewer of them are going to meetings. More newer generation, or really all of us, are looking toward social media to learn new techniques, and so all of our investigators are active at all sorts of levels of social media. Just an example of a podcast that I did with the Osteology Foundation on regeneration, and 2 podcasts that done last year that relate to this. One with Sam and my partner talking about PDGF and how we use it clinically, and the other one with Dr. Wu in Harvard and Dr. Gianobili and Irina's in the audience, and Jeannie with the H-Credent program, where it's a NIDCR sponsored program to help teach junior faculty members and dental students how to do, or the importance of practice-based research. So it's important for these podcasts to be disseminating information as well. But you can't do podcasts every day, but you can do other forms of social media, whether they be TikTok or these are Instagrams. Again, pushing people toward publications, toward meetings, toward whatever you're trying to disseminate. We're also very keen on making certain that our publications, this is all part of the dissemination process, that we need our publications being top of mind, being out there. And we're proud that out of all of our publications from our network, we have we've been awarded 11 major awards for our publications. Each year, the American Academy of Periodontology awards 1 clinical research award and 1 Robinson Regeneration Award, and they are supposedly represent the papers that are the most clinically relevant of that year, and we've have 11 of those. So the professional recognition is very important. But I almost think that the peer recognition may be even more important. And the peer recognition is where, we see, as it's on the slide there, the most downloaded paper at a certain time, or 500 views. All of these things mean a lot. And to me, they probably mean more than the professional recognition, although I'm pleased with that. But to be honest, the professional recognition is awarded by a group of so-called experts that are appointed. I think of it as kind of like a dark room with cigar smoke, and there shouldn't be, but there's personal and professional jealousies. Corporate influence sometimes seeps under the door, but this peer recognition is really important because it shows what all of us are really interested in. And then finally, public recognition. This is in 2007, we did an RCT using the patient's own fibroblast to try to close open interproximal spaces. That year, that was the most downloaded paper for Journal of Periodontology. And on YouTube, we've received over 13,000 views, mainly from the public, because there's a lot of public interest in this. And so that's pushing, it's another way to look, because here we're pushing it from the public, toward us rather than us toward the patient. And that, of course, is one of the reasons why you see all these drug companies advertising on TV. So dissemination's an important thing. just as important is practice-based research is able to capture patient-reported outcomes. You know, that sounds easy, patient-reported outcomes. You know, what's the big deal? You ask a patient, did you like it or not? And it's not that at all. And to capture meaningful ones, it takes a lot of knowledge and methodology and know how to do that. And we're able to do that. And why is that important? Because if we're looking for alternatives, it is unlikely we're going to find an alternative to the gold standard that is as good or better if you're looking just at objective measurements. And so most alternatives to gold standards are going to be chosen on something that's almost as good, but the patient-reported outcomes push it over. That's why patient-reported outcomes are so important. In addition to that, we're able to capture clinician-reported outcomes. And this is where alternatives may actually outperform gold standards. You know, how much does it cost? How easy is it to do? Do we need an extra auxiliary? Those kind of things are important also when we're looking at alternatives. You know, today, it's not just about the data. It's about how the procedure feels to the patient, how manageable it is to the clinician. These factors may ultimately determine adoption more than clinical endpoints. A big difference in how it used to be. You'd live or die on tenths of millimeters, and how that felt to the patient or what it cost made no difference. We also work with people around the world. And that's important for a number of reasons. If we're working with a company, you know, most of them, they're anxious to get to North America because that's the biggest market. And if you're going to get to North America, you have to have studies done in North America. You have to have your studies published in North American journals. On the flip side, we can do studies that will satisfy European regulatory matters. So sometimes it's easier, more efficient for a European company to do a study in the US, and we can go back and forth. Yes, we are a practice-based research institute, but we also work with universities. You know, we commonly work with leading universities that see our work as synergistic to what they do best. You know, it's common for us to work with universities where we need very sophisticated imaging or biomarker analysis and those kind of things. Also, you know, we'll work with universities on multi-center trials. So, you know, it's by combining the rigor from the academic centers with the reach in private practice that generates data that's credible and scalable. We also have a registry, which I think is really cool. On a couple of different levels. I mean, everybody remembers the evidence pyramid, okay? You got, you know, your RCTs and stuff on the top and case series and that kind of thing on the bottom. We did a study 3 years ago where a sponsor came to us with a new implant, wanted us to do an RCT with a new implant, but also look at their workflow. And so we created a highly controlled RCT, 100 patients, 9 investigators across the United States. All the investigators are master clinicians. I've tried to make the argument that if you do a study in a practice-based environment, the results should be generalizable to the practice. Well, in parallel to that, we have a registry. And in that registry, in round numbers, we have 2,000 implants, of these kind of implants, and 700 dentists from all over the world. If what I just told you is true, the results should mirror each other. And we're just now in the process of beginning to analyze that data, and you'll be seeing some publications start coming out in the next year or so. And also the registry allows a conduit for other study sponsors to be able to look at their products in a fairly economical way. Brings lots of great things. And we're also involved in artificial intelligence. I mean, today everybody has to be. I mean, we use artificial intelligence to help us streamline protocol development and all sorts of things. We published in the Journal of Periodontology last year our first paper using AI to improve compliance of our patients. We're working right now with another AI company on a study where we're creating decision support systems to help us better understand risk. You know, you just have to jump on the AI bandwagon. I mentioned yesterday in my lecture, I don't think you have to worry about being replaced by AI, but you may be replaced by somebody who knows how to use AI. So it's important that we all incorporate that as we move forward. So in conclusion, yes, we're driving clinical therapy towards successful adoption. It's not easy. There's nothing easy about it. But we do it by producing the real-world evidence that the FDA and we demand, producing meaningful data and long-term data that are important to clinicians and patients alike, that are validated by pros and ClinRos, and then disseminated by key influencers, allowing us to really look for that next big thing, to collapse that translational lag, can't be 10 to 15 years, and to be able to find that next big thing that's really going to provide real impact in our patient's life. Practice-based research is redefining what's possible in research, and it brings all of these unique things that I just mentioned. And it's a process, and without all of those things, it's really difficult to get from that promising early results to the FDA approval all the way down to being widely adopted at the clinic. So thank you very much for your attention.
We can do a little Q&A session. So I have a question actually for Dr. McGuire. So where do you think the future of oral regeneration is going? Maybe one or two trends that you're seeing in the field, and to shed light on that.
You know, it's really exciting. On one hand, I'm a big believer in growth factors, and I like I like to work with active, not passive devices. But on the other hand, there are some matrices that may have the ability to use biomimetics to signal natural functions and using the body's own growth factors and that type of thing. And I'm fortunate to be able to see a lot of this new stuff that's coming in. And although I've been in practice a long time, I'm more excited now than I've ever been. So I think that I have a traditional periodontal practice. By that I mean we do implants, but my job is to save teeth, and so I would have a hard time, I think, convincing my patients to allow me to do, just an open flap debridement or something like that without the hope of regeneration. And I think that I can provide that hope and be honest with the fact that we should really be able to help them. And there's no reason that in today's world that most of our patients can't keep their teeth for their lifetime.
Great, thank you. And for Dr. Lynch, where do you see the future of oral regeneration headed towards?
I think Mike's much better positioned than to talk about oral tissue regeneration. I would say, David, regeneration as a category in medicine is booming and just in its infancy. I think some of the greatest advances in medicine in general will be in the regenerative medicine space, whether it's in the oral cavity or brain. I mean, we haven't even begun to understand how to enhance healing or let alone regeneration in the central nervous system. And that, I try to convince my kids, you should go into that area because that is, I think, one of the incredible frontiers of medicine.
Great. Thank you very much for your insight. And I think the final takeaway is it's really exciting for us to be in the field of periodontology, and I welcome every single one of you to get involved. Whether it is practice-based research, it is publishing in your private practice about case reports, case series, or contributing to the path of scientific discovery. So we are all one community, and we can have all unique ways to give back to our specialty, our profession, and to the field of dentistry. So on that note, thank you very much for your attention, everybody, and have a great rest of the conference.